Assalamualaikum and hello. My name is AMIRAH FARZANA binti ISMAIL, and I'm the representative for my group, which is group 5. So these are the members of the group. Let us move on to the question number 1. So this question basically asking us to explain the experiment and the stuff involved in cDNA microarray, as well as to confirm that the identified genes are really involved in the production of the cell. So in order to identify the genes involved in the production of taxol in Texas brevifolia, we need to do a cDNA microarray. The first step in the experiment is we need to collect the sample from the tissue or the cell that we are interested in. So in this case, it's the bulk of Texas brevifolia. Next, we divide the sample into two, which are sample A that will be treated with methyl jasmonate and sample B that will be untreated with methyl jasmonate. Then mRNA is isolated from both samples. To create a cDNA, reverse transcription of the mRNA is done. Both samples are then incorporated with different fluorescent dye. So sample A will be treated with red dye, while sample B will be treated with green dye. This helps us to distinguish the sample category. Next is the hybridization. The sample are placed in the DNA microarray so that each cDNA gets hybridized to its complementary strength. And lastly, the computer scanning for analysis the result and collecting the data. From the result, we can see that the red one is gene expressed in the methyl jasmonate treated culture. The green one is the gene expressed in methyl jasmonate untreated culture. And lastly, yellow, the gene expressed in both culture. From the result, the example of the genes that were identified to be upregulated in the microarray can be seen from the slide. So how do we confirm that the identified genes are really involved in the taxol production? So our group suggests to use the T-Coffee program, because this program is one of the most efficient programs for multiple sequence alignment, because they allow us to detect the conserved domain of the sequence. Thus, the conserved domain can tell us that those sequences are crucial for cellular stability and the function of the studied genes. T-Coffee programs are also applicable for both protein and nucleic acid sequence, and it helps us to save more time to compare and detect the genes. Moving on to question number 2. So this question asks us to explain the experiment and step involved in the quantitative real-time PCR, and to consider the detection method that we get with each the TAQMAN probe, as well as to provide their advantages and disadvantages, and the difference, to explain the difference between the result obtained in the Q real-time PCR and the normal PCR. So qPCR is also known as real-time PCR. It is a method that helps us to collect data through a PCR process by combining the application and the detection steps simultaneously. In this method, RNA is first transcribed into a complementary DNA or cDNA by reverse transcriptase from the total RNA or mRNA. So the cDNA is then used as the template for the qPCR reaction. qPCR is mainly performed with TAQMAN probe, which work with probes specifically designed to bind the sequence of interest upon synthesis of the cDNA strands. The probe is cleaved, which results in a fluorescent signal. In TAQMAN assay, the strands separate in the denaturation step. After this, the primers can bind and the TAQMAN probes will be specifically bind in between the target sequence. The top DNA polymerase has a 5' exonuclease activity, which will chew away the target specific probe. Cleavage of the TAQMAN probe separates the reporter from the creature, which leads to the emission of fluorescent signal that can be detected after each cycle. Then the process will be repeated before analyzing the PCR product. So this based on what I explained before about the TAQMAN probe. So TAQMAN probe is principally relies on the 5' to 3' isonuclease activity of TAQ polymerase and fluorophore-based detection. We are using the qPCR method to quantify the TAQMAN probe because qPCR measures the intensity of fluorescence emitted by the probe after each cycle. So the resulting fluorescence signal from the TAQMAN permits a quantitative measurement of the accumulation of the product. So what are the advantages and disadvantages of TAQMAN probe? So TAQMAN probe are the most popular, and it can increase the specificity of the qPCR, and then it allows us to multiple detection of PCR product in the same sample. While the disadvantages are it's usually high cost and the synthesis of different probes is required for different sequences. So what are the difference that we can find in the result between qPCR and PCR? So qPCR has a high resolution and it takes shorter time compared to PCR, which is approximately between 1 to 1.5 hours to complete. And the result obtained during the reaction, we can obtain the result during the reaction, which is during the exponential phase. While PCR, it has a low resolution and it can be time consuming, and the result can be only obtained at the end of the reaction. So I think that's all from my note. Thank you.